Glucose, product of photosynthesis. Type of carbohydrate. Quick fuel and short-term energy storage. Common monosaccharide. A simple single sugar, 3 to 7 carbon, particularly in the body. All cells burn glucose to make ATP, energy to meet their immediate energy demands found in blood. Maltose. Glucose undergoes dehydration, requiring the enzyme maltose to become the disaccharide double sugar, called maltose. Maltose equals 2 glucose plus H2O. Maltose is table sugar. Sucrose is table sugar, which equals 1 glucose plus 1 fructose plus H2O. Lactose is milk, which equals glucose plus galactose plus H2O. Fructose, which is fruit, sugars, galactose, and glucose are all isomers of each other. They have the same chemical formula, which is C6H12O6, but different atom arrangements. Polymer of glucose is polysaccharide, highly branched. Storage form of glucose in animal chains of glucose have many side chains compared to starch. Liver converts glucose to glycogen for storage. In between meals, liver releases glucose into blood concentration, and it remains at 0.1%. Starch is a polymer of glucose, polysaccharide. It's a storage form of glucose in plants. It's fairly straight chains with few side branches off the main chain. Forms from dehydration synthesis between many glucose molecules. We eat starch in the form of potatoes, and it is stored in our body as glycogen. Glycerol is a neutral fat. It's the backbone of fats. In other words, it's a triglyceride. It has 3 hydroxyl groups responsible for solubility and the ability to absorb moisture. Fatty acid is a sequence of C and H with a carbolic acid at the end. Saturated fats are produced by animal and solid at room temperature. In other words, or for example, butter. Unsaturated fats are produced by plants, tissues, and liquid at room temperature. Example, vegetable oil. Neutral fat is produced by dehydration synthesis of one or more fatty acids with glycerol. It results in monoditriglycerides. Can be formed with saturated or unsaturated fat. Don't mix with water. Non-charged, non-polar molecules. It's used for padding protection and long-term energy storage. Phospholipid is important components of cell membranes, have the same basic structure as neutral fats, except one fatty acid is replaced by a phosphate group with a charged nitrogen attached. They have a phosphate containing a head that is hydrophilic, so likes water, and long fatty tails that are hydrophobic, which means doesn't like water. Hydrophobic is also nonpolar. Protein organization. Primary structure is a structure of amino acids joined together by the peptide bonds. There's 20 amino acids, which equals millions of sequences. Secondary structure, since peptide bonds are polar, H bonding routinely occurs between amino acids in the primary line. This chain will coil up in the shape known as alpha helix layers, called B-pleated sheets, that also form. Tertiary structures are different types of bonds between R groups. They make alpha helix bonds and turn forming globes of protein of all shapes. The bending causes new bonds to form, creating a 3D shape. Quaternary structures. For proteins with more than one polypeptide chain, the quaternary structure is the specific arrangement of polypeptide chains in that protein. ATP is a type of nucleotide that is the primary carrier of energy cells and consists of sugar, ribose, base, adenine, and 3 phosphate groups. Bond between 2 outer phosphates is very high in energy and needed for reactions. When broken, lots of energy is released. ATP is produced mostly inside mitochondria during the process of cellular respiration. When the bonds break, ADP and a molecule of inorganic phosphate form.